Dr. Padma. Dr. Padma Devarajan is author and chair professor, SIES School of Pharmaceutical Sciences. She is retired recently as senior professor in pharmacy and former head Department of Pharmaceutical Science and Technology, former dean research and innovation at ICT Mumbai. She was also member of board of governors of ICT Mumbai. She has supervised over 100 students for the master's and PhD degree. Her research is in the field of drug delivery system and diagnostic for human and veterinary healthcare. She has over 100 publications, many granted patents, and has licensed technology to industry. And some products are already commercialized in India and Europe. She is editor and author of 2 books on targeted drug delivery system published by Springer. She is a only Indian academician who was on the board of scientific advisors of Control Release Society USA. She represents India in ISO workshop, working group on health, safety, and environment aspects of nanotechnology. Is on the editorial board of national and international journals, and also consultant to the pharma industries. She is a nominated fellow of Maharashtra Academy of Science, currently president of Society of Pharmaceutical Dissolution Sciences. She is a recipient of many awards for research and innovation, some being Outstanding Woman Scientist Award of ICAR and Woman Scientist Award 2024 of Control Release Society USA. Over to you, ma'am.
A very good morning to one and all. Let me start by thanking the organizers, and in particular Dr. Kedia and the team from IIT Mumbai for inviting me to give this talk. Lipidic nanoparticles have come like a bang into the scene since COVID-19. So I think it's very apt to see whether they could really go a long way forward in nanotechnology. So I'm going to talk to you about lipids in nanotechnology for infectious diseases, more through case studies and a little bit of theory. But more importantly, when we talk of nanotechnology, the whole world talks of cancer and nanotechnology, and the focus on infectious diseases is very limited. Like you can see here, we've been talking of liposomes all morning, and I've listed just 4, but there are many more, but in the cancer space, very limited in the infectious disease space. And one reason that I think is a cause for this kind of paucity in infectious diseases is because these infectious diseases afflict the lower social economic strata and may not be beneficial for the pharma industry. So we as a research group have been trying to pick out such areas and develop, and we're also trying to take it to the market. So what I would be talking today is about targeted drug delivery for infectious diseases. And when we talk of infectious diseases, generally we can classify them into 3. We have the extracellular, we have the intracellular, and we have the mixed. But nanotechnology is more relevant for the intracellular infections, particularly because nano can target the reticuloendothelial systems and be taken up readily by the macrophages. And therefore, it has relevance for the intracellular infections. So what we're going to talk about today is nanotechnology for infectious diseases, combining them with the advantage of lipids in nanotechnology. So the whole concept of nanotechnology, targeted delivery, etc., started long ago. It was way back in 1898 that Paul Ehrlich proposed the magic bullet or proposed targeting by using bodies which possess a particular affinity for a certain organ as a carrier. So that actually converts to drug lipid conjugates, but today it has translated beyond that to a magic wand where you have the carrier material in which you entrap the drug, don't just physically complex it. So you have the carrier defining the biodisposition of your system, and therefore you can take the drug to different places in the body. much later, you had Richard Feynman, who brought in the concept of nanotechnology in the 1900s. And together, you had the emergence of nanotechnology, which is being widely used. And when I talk of nanotechnology, it could be passive targeting. It could be even active targeting. But what's important to understand is lipids can be used for both passive and active targeting. So they are versatile, they can be used in both places. And why are we looking at nanotechnology for infectious diseases? Because we have the reticuloendothelial system, which is a general body defense system, which can engulf and destroy foreign substances. So we rely on the basic physiology to entrap particles the way it would entrap any foreign body. And here what you're seeing in these pictures is how the entrapment actually occurs. This is more like a cartoon, but this is actually a live demonstration of macrophages actually taking up particles. And the major cells for the intracellular infections are the macrophages, which are present in the liver, the spleen, the lungs, the bone marrow as a major organs, which are the fixed macrophages. We also have the WBC, lymphocytes, etc., which are our floating macrophages. So now let's come to nanomaterials for nanomedicine. One huge group of nanomaterials are the polymers, with which you can prepare a range of systems like particles, capsules, micelles, dendrimers, depending on the nature of the polymer. And the next set of materials are the lipids, which is the focus of today's seminar. So under lipids, we already had Dr. Kedia showing a range. So you can see you have a range of lipids. You have natural, you have synthetic, you have semi-synthetic, and you have also a range of formulations. Other than liposomes, you have SLNs, you have nano-emulsions, and you have nanolipid carriers, and many more. I have not been listing all of them. But what I would be focusing on is solid lipid nanoparticles. Liposomes has been the focus till now, but the reason for focusing on solid lipid nanoparticles is because of our objective is to bring down cost. And while we focus on cost through excipients, although we are not engineers, we've also been focusing on bringing down the process challenges and making the process very simple, as you will see in my later slides. So first, since it's a lipid-based seminar, what are the advantages when I have a lipidic nanosystem? Lipids are biodegradable and the body is designed to metabolize lipids. So many lipids could be safe, and that's what we are trying to use, the safer lipids. They would be biocompatible. They're versatile because we've seen you have so many types of lipids from which you could choose. Toxicity is low because they're likely to be biodegradable and biocompatible. They are amenable to green technology, which is again a very important aspect because you're not using solvents, you don't need to, damage the environment. And with the lipids, you can also get improved drug stability and protection. So you have various applications. So this tells you this is the versatility of lipidic nanoparticles, right from targeting cancer, vaccine delivery. We've seen all this briefly in the morning session. So now let me run you through two examples where we are actually showing application of solid lipids which could be translated. So first, I'm going to talk to you about the AMB is amphotericin. So it's a lipidic nanosystem of amphotericin B, and it was the PhD work of Dr. Pooja Todke, who completed just 2 years ago. Now, the biggest challenge with amphotericin B is, although it's a gold standard for visceral leishmaniasis and also mycosis, which are deep fungal infections, which is rarely mentioned anywhere with emphasis, the conventional AMB nanotherapy gives a lot of challenges. It's highly toxic to the liver. It's got very high kidney toxicity and it's low cost, and therefore it still continues to be used because there is no option. When you look at liposomal AMB, it's safe. I think, okay, yeah. But In case of the conventional, you have it associated with fatal kidney toxicity, which could be 5 to 10%, which is very high. It's a huge number. And with liposomes, we've seen different methodologies and simplification of methodologies. So there's a high cost, there's a scale-up challenge, and particularly for amphotericin B, the availability, even for people who could afford to pay, was a challenge. It was not available because of the scale-up challenge. So we are looking at a safe and affordable amphotericin system. So where did we begin? We began by screening lipids. I'm just showing you 2 of them, but we've screened about 10 to 12 of them by first docking and then by molecular dynamic simulation using MOE software. And this is just a brief example to tell you. In case of amphotericin, the efficacy is because it interacts with ergosterol, a fungal lipid, and the toxicity is because it interacts with cholesterol, a mammalian lipid in the human system. So the method that one needs to adopt for a safe system and efficacious is to eliminate the interaction with cholesterol and accelerate the interaction with ergosterol. So we are looking at lipid 1. The red arrows show you that it is not interacting. That's why I put a cross there. And the green arrows show you that you do see an interaction with cholesterol. If you look here, oh, this is literally running. I'm not able to control this. So if you see here, there's extensive interaction. Now, if you come back here to lipid 2, you will see that it does not have interaction with ergosterol. So it's not likely to be efficacious. And it has interaction with cholesterol, so it's likely to be toxic. So this way we were able to screen out, and we did see some lipids which interacted with neither or some that interacted with both. So all those we screened out, and I'm just showing you 2 lipids at 2 ends. So we are now looking at the liposomal technology. Because like I told you, it's not only a lipid that we chose, but we also came up with a very revolutionary process to prepare these solid lipid nanoparticles. So this is a step to prepare liposomes, which has been simplified as Dr. Mahesh showed us. This is a conventional method to prepare solid lipid nanoparticles. So you have a lipid dissolved in an organic solvent, or you have it melted. So you have a molten, in which case you make it green technology, you bypass the solvent. Then you precipitate, you homogenize, solvent removal, isolation, lyophilization, and particularly when you come to the last 2 steps, there's a huge chance of agglomeration. And that is again a huge challenge that needs to be bypassed or circumvented. But now I'll take you to our technology. We call it in situ nanotechnology. So the nano system is prepared at the patient bedside. It's not prepared by the manufacturer. So what does the manufacturer supply? The manufacturer supplies a simple pre-concentrate, which is a solution. And we all know how easy it is. We have a lot of people from industry here who know how easy it is to make a sterile injectable solution. So it's a sterile injectable solution of amphotericin plus excipients in an injectable solvent. And we just filter it. We get a sterile pre-concentrate filled under aseptic conditions. So this is the sterile pre-concentrate. So you can see that it's a simple process, it's scalable, and it's also affordable because the process, there's no challenge. It's a low-cost process. And if I look at my lipid, it's again a low-cost lipid when I compare the price with phospholipids, which are used in liposome. Now I will demonstrate to you this process. So that pre-concentrate is withdrawn. Because seeing is believing, so you can actually witness this process. Now we have instantaneous generation of the AMB loaded, and this is ready to inject. 95% of the drug entrapped. The size confirmed by TEM is about 80. The PDI is slightly higher. Normally 0.3 is a cutoff, but we are getting a little over 0.3 which is 0.34, which is perfectly acceptable because of the simplicity, the reduction in cost, etc. Now with AMB, we also have one challenge of it being in the aggregated state. So this UV spectrum with a peak at 322 tells us that it's not even in the aggregated state, it is in the safe monomeric state. Actually, this is not monomeric, it's a super aggregated state, but the super aggregated state releases as monomers, does not allow aggregation once it is injected. So now we did all the other studies. So I'll run you through some safety studies quickly. We did a hemolysis study and we have compared everything with liposomal amphotericin B, which is the red bars. And if you see, I direct you only to this one bar here. The third bar from here, the bar next to the liposome is our final product, which we consider optimal. So if you can see, hemolysis is very low. The renal biomarkers, because we saw it could cause fatal kidney toxicity or renal toxicity and hepatic toxicity. So if you look at the renal biomarkers, you find that our system is very comparable with the liposomal system. We looked at histopathology, and again with histopathology, we got this analyzed by a pathologist, and we were told that there is no significant change, no significant signs of toxicity. We then did the in vitro anti-leishmanial activity. This was done at CDRI, and we used the macrophage amastigote model. So we had the amastigotes entrapped in the macrophages. Again, this system was compared with liposomal AMB, which is the red. And if you see the IC50 value, the green is our system. We see that the IC50 is twofold lower than the liposomal. And in case of the specificity, it is comparable to liposomal. So we believe it could be said that it is comparable to liposomal. We may not really call it as something more efficacious, but we have something that is comparable to liposomal. We've done also an antifungal assay because I said we're looking at mycosis also as one of the treatment opportunities for the system. So even in case of this fungal, both the assays, we found our system comparable with liposomal AMB. So we have a system that is highly promising. We are now moving further. We've already done a brief toxicity, which is not a GLP toxicity, so I have not put that data, but it tells us that it is safe at 25 times the proposed dose, but we are planning to do a complete GLP toxicity, and this patent got approved just yesterday. Yesterday night, I got a message from the agent that this patent is approved. So that's why it's not put up on the slide. So I'd like to run you through one more system if I have the time. Do I have the time?
Yeah.
5 minutes?
Yeah, 5 minutes.
I'll finish in 5 minutes. I'm going to go really fast because I'll do it like a Jhalak Diklaja. Okay. So we're looking at another big area, and that's the veterinary. And then veterinary, whenever I've been talking to the people in the industry or the doctor, they say, you can't have liposomes. They're very expensive and not affordable. So can you develop something cheaper? So that's where we began. And we're looking at zoonotic infections and other difficult to cure infections in animals. And when I say zoonosis, it's even more critical because these infections come to man through products and through being with those infected animals. And such infections, you have brucellosis, which is now being declared as a national kind of endemic. So currently what is done world over is slaughter of these animals, but in India, slaughter is not permitted because of socio-religious compulsions, and therefore treatment opportunities are being looked at. We were approached by the vet college and we decided, so this is solid lipid nanoparticles or buparvacone, a drug used for theileriosis, which is a veterinary infection infecting cattle, and it significantly influences milk production. And these are crossbred cattle developed to improve milk production. So there is a significant effect on the milk productivity. This, what I'm showing you, is a proposed product. This is licensed and the patent is granted and it's being developed. The permissions from DCGI are obtained for manufacturing, and the manufacturing is beginning in Dehradun. So this was the PhD work of my student Hina. Now here I'm just showing you, it's the same thing. We prepared a buparvacone pre-concentrate, 20 ml. we dilute it into 250 ml of dextrose injection. So we're taking it in the second picture. In the third, we are adding, and you can actually see the nanoparticles forming. And these nanoparticles showed us an entrapment efficiency, again, of greater than 95%, if you see it in this last one. The size was about 600 nanometers, but we have seen that up to an average of 600 to 700 nanometers also we can administer. We have done studies in cattle with the medical people. So we also did a biodistribution, and we saw high accumulation in the RES organs. This is an infection, intracellular. So this was the first step. And then we actually quantified the scintigraphy in each of these organs. And if you see, the tallest bar represents the spleen, and then the next represents the liver. So we are seeing very high splenic uptake, which was not expected. And here we have another paper published based on why we got splenic uptake, because it was due to the shape of the particles not being round. But theileriosis is a splenic infection, so this became an unexpected advantage for us. And then we have actually looked at toxicity. And here you will see that the MLD, the maximum lethal dose, was greater than 25 milligram. And what we are going to inject is just 0.5 milligram. So again, very high safety that we've seen. Now, we did a clinical study in collaboration with a college in Akola. These are the clinical manifestations. You have lacrimation, you have enlargement of the lymph node, you have the paleness of the vaginal mucous membrane and opacity of the eye. So these pictures are the clinical methods of diagnosis and evaluation of the symptoms. We've also done PCR to confirm whether it's working. Now, fever is something that is one of the symptoms of theileriosis, and we see that fever has gone down. The very crowded slide, and I'm not able to use the pointer, I'm sorry. So we saw that there was a significant reduction in the temperature. And more important, I'd like to take you to these two. 0.5 was the liters per day during the infection, and after treatment, it went to 10.8. Similarly, 1.5 went to 12.4. Here, this cattle stopped milking and it went to normal. So this was, again, a very important indicator of efficacy. So all this was done in collaboration. This is a total collaborative study. And more important, since this was a targeted system, it was not seen in the milk. The limit that FSI puts for the residue in milk is 10 nanogram per ml. And when milk was analyzed, we saw that it did not exceed 6 nanogram per ml, which means during treatment also milking could be continued. And this is very important for the small farmer, whose daily income depends on the milk that he is collecting from his cattle. So no withdrawal time was again one very important advantage of targeting buparvacone to the nanoparticles. So when we talk of lipids in nanotechnology, they have a huge role as carriers. And for infections, their hydrophobic nature plays a very important role in enabling high RES uptake. And therefore, for intracellular infections, lipids could play a very, very critical role. So what I would like to acknowledge my collaborators, Dr. Abdul Samad, who's a retired dean and director of MAFSU, and Dr. Gaikwad, who's soon going to retire. These are earlier studies. This is the institute where I was until end of April, the Institute of Chemical Technology. And this is my vibrant research group, because of which we keep doing a lot of good work. And this is where I am now, the SIES School of Pharmaceutical Sciences, where I hope to continue this work and bring in the ICT tradition into SIES. Thank you so much.
Thank you, doctor. It's really inspiring to know that, how concept to be converted to the product which will be useful for the society. Yeah, thanks for comprehensive presentation. This is question and answer time now. Yeah.
A very nice talk. Thank you. I'm wondering if you have thought about the process of encapsulation that is taking place. How is that so you have this initial mixture which has lipids, maybe an organic solvent, I do not know. No,
No,
so
it's a ready to inject.
it's ready to inject. But then you,
Finally, so there's no organic solvent. Absolutely no, because then it's not ready to inject. I have to remove it, right?
yeah, but then you dilute it with something, right?
With dextrose.
Just the dextrose.
I am putting it because that's a standard method. In case of cattle, if it's a big injection, it's diluted with either saline or dextrose saline or dextrose. Now, in this case, we are using dextrose because we don't want the sodium chloride charge effect to affect our nanoparticle formation.
Okay.
So, we've kept dextrose for isotonicity. Because I cannot inject anything that's
Absolutely,
not isotonic.
Yeah. So these are lipid nanoparticles, right?
Solid
These are
lipids.
Solid
lipids. And approved.
Yeah.
One of them is approved. The other one is not approved. But what I showed you, buparvacone, we've done the entire studies. In fact, we've even done the genotoxicity and development toxicity and all of that lipid and shown that it is safe. And that's how they got the approval.
Okay, thank you.
The formation of the SLN basically is happening like you showed in the video.
Yes.
When you are injecting through the pre-concentrate, when you are injecting, right? So how do you control the precipitation, like to define the final particle size?
So one thing here is we have been instructing that they should continuously inject without stopping and touch the needle into the solution and do it. Till now, it's all been controlled, the studies, the clinical studies. But actually, the theory here is very simple. It is simply nanoprecipitation.
Right.
Now, just because you do it in a beaker, it's science. But the same science we've extrapolated here by using everything that can be injected. That's all I've done.
Yeah, so I was just thinking
it's the same science of nanoprecipitation.
Right,
so we have publications also. And in fact, for buparvacone in particular, we got a very different asymmetric shape. And that gave us a splenic uptake. So we have a fantastic paper on why the asymmetric shape happened. That also we have a paper.
So my thought was, since you mentioned there's a 600 to 700 nanometer, which is though efficacious, if there is a need to reduce it, is there a way to control the injection velocity or the volume?
We can try, but then this was done repeatedly and we could get this only. And it was safe. We have done a toxicity in mice also. So it is safe to inject. Absolutely. And technically also, if you look theoretically, anything less than 1000 nanometer is considered safe to inject, although nano doesn't define this as nano. Typical nano doesn't define this as nano.
No, that's fine. I just wanted to understand mechanistically.
This is an application of larger nano for nanomedicine.
Yeah, fine. Thank you very much.
Yeah, I think. Any further questions? Okay. Thank you, ma'am.
Thank you so much.
I will request, just a minute. I will request Professor Samir to give the